introduce Professor Geeta Narlikar. She is a professor of biochemistry and biophysics at the University of California, San Francisco. She obtained her MSc in chemistry from IIT Mumbai, PhD in chemistry from Stanford University, and postdoctoral research was at Harvard Medical School. She has been a faculty member in the Department of Biochemistry and Biophysics at UCSF since 2003. She studies how the folding and compartmentalization of our genome is regulated to generate the many cell types that make up our body. Her laboratory has pioneered the application of sophisticated biophysical approaches to study the mechanisms of macromolecules that regulate genome organization. Through these studies, they are learning how nanoscale molecular motors use chemical energy to cause mechanical disruptions in the packaged genome. That the smallest unit of genome folding, a nucleosome, acts akin to a dynamic receptor rather than a static packaging unit, and that liquid-liquid phase separation processes can help organize and sequester large regions of the genome. Pioneering discoveries from her laboratories are changing textbook descriptions of genome packaging and suggesting new avenues to tackle diseases caused by defects in genome organization. She enjoys teaching and mentoring graduate students. She believes that kindling the fire of curiosity within graduate students and consistently supporting their initiative brings out the best in them. In addition to tackling apparently insurmountable questions, she is an avid mountaineer and has hiked up the Sahyadris. She has many awards to her credit, including the Beckman Young Investigator Award, the Leukemia and Lymphoma Society Scholar Award, the Outstanding Faculty Mentorship Award by the UCSF Graduate Student Association, Glenn Award for Research in Biological Mechanisms of Aging, as well as she is a distinguished alumnus of IIT Mumbai. I take this opportunity to invite Professor Geeta Narlikar to deliver her talk on the dynamic genome.
Thank you, Kundan, for that wonderful introduction. While I mic up, I just want to thank everybody at IISER for hosting me and my family for the last almost 6 weeks. We've been here doing various things in IISER and outside IISER, including teaching a course at IISER. And I have gotten to know the faculty and the students at IISER in this process. And it's truly a wonderful atmosphere to do science and a really great context and environment to learn. And so I just want to thank the whole IISER community for being such wonderful hosts for my family and me. And also for the invitation to give this talk today to all of you. Great. So I changed the title a little bit. When I gave this talk at IIT, I had given the title I had given to IISER. And the public relations office at IIT said, can you make the title a little more catchy? We want to get the BTechs to show up, the engineers to show up. And so this is the title I came up with. But clearly, people in IISER don't need a catchy title to show up. Okay? So you got the old title, and you still showed up. So thank you. But this title might more accurately represent what I'm planning to share with you. And I want to start by acknowledging that the human genome has captured human imagination for almost the last 20 years, ever since the first draft sequence of the genome was obtained for humans. Okay? And so about 20 years ago, the first draft sequence of the human genome was obtained, and that led to headlines such as this in the New York Times, that the genetic code of human life is cracked by scientists. There was a lot of excitement, and rightfully so, because as we've all learned in textbooks, the DNA sequence in our cells contains the instructions for life. The DNA sequence in each one of our cells tells a particular cell how to behave, tells a heart cell how to behave like a heart cell, and a brain cell how to behave like a brain cell. Okay? And so since the last 20 years, since the human genome has begun being sequenced in depth, we have experienced many benefits from sequencing of the human genome, all the way to the current day and age, where even in common consciousness, we can now talk about genetic tests. Okay? We can talk about tests where the DNA of an individual, like any one of you sitting in this audience, can be taken through spit cells, through spitting in a tube, and sequenced by various companies. And by sequencing pieces of your DNA obtained from your spit, these companies can tell you your chances of, let's say, getting breast cancer or ovarian cancer, or as 23andMe does in the US and in other countries, your risk factors for developing diseases like Parkinson's or Alzheimer's. Okay? And so many of you may know somebody who's done these tests, or you may have at least heard of these tests, where all you have to do is spit into a tube, send your spit off by mail, and they send you your risk factors for various diseases by sequencing parts of your genome. Okay? And this is something I found from a couple of weeks ago, that these genetic tests are also gaining popularity for diagnosis and treatments in India. And there are many companies being set up in India over the last several years to use the information from genome sequencing and to use the technological advances in genome sequencing to help the general public get a better sense of their disease risk through sequencing individual people's genomes. Okay? And so it's not a surprise that the human genome and the sequencing of the human genome has captured people's imagination, the common person's imagination. But what I want to emphasize today and throughout the talk is that these kinds of benefits to society rely on first discovering how nature works. Okay? So discovering how nature works is essential to derive benefits from its workings. Today, we can derive benefits from genome sequencing because for more than 2 centuries, scientists have been curious about how the genome is organized. Okay? So while we've been excited, or the common public has been excited over the last 20 years about sequencing the human genome, it's important to keep in mind that these discoveries and these benefits arise from the work of curiosity-driven research. And these benefits arise from the work of many, many curious scientists over many centuries. Okay? So in the context of genome sequencing, where we are now is because we are standing on the shoulders of many scientists who are curious about what is this material inside the cells. Okay? And so starting from the late 1800s, where chromosomes were seen in the nuclei of cells, to early 1900s, where chromosomes are carriers of heredity, were discovered to be carriers of hereditary information, all the way to genes being discovered as drivers of embryonic development, to a double helical model of DNA in the 1950s that is in textbooks that almost everybody in this room should know about, the double helical structure of DNA, to the current day capacity to sequence the genomes of individual humans. Okay? Now, all of these findings and discoveries have built on each other and have, again, built on a curiosity-driven basic science. Okay? And so let's come back to the current day, the capacity to sequence the genome of individual humans. And let's ponder where we are and what this might spur us to think further. Okay, so let's come back to these tests. And let's think about a little bit what this means. You spit into a tube. You send your spit cells off to somebody to sequence. Okay. Now, somehow, by sequencing the DNA in your spit cells, people or we can figure out, or the scientific community can figure out, they can't figure out how this would work, but okay. So somehow by sequencing the DNA in your spit cells, you can get an understanding of how your brain cells or your ovarian cells will misbehave. Okay. Step back for a minute and ask yourselves, how is that possible? How is it that information from a spit cell will tell you something about your brain cells or your ovarian cells? And somebody in the audience would say, of course, we've learned in high school, or perhaps in IISER, depends, maybe in IISER, we've learned in IISER that the sequence of DNA in all of our cells is the same. Okay? So the DNA in our spit cells should be essentially the same as the DNA in our brain cells or the DNA in our ovarian cells. Okay? And so if the DNA is the instruction for life, contains the instructions for life, then it doesn't matter where you get the DNA from, whether you get it from spit cells or heart cells or blood cells, it's still going to give you information about life. Okay, so that's fine, but that leads to another conundrum. If the DNA sequence contains the instructions for life, and if the DNA sequence contains instructions for what makes a spit cell and what makes a brain cell, and if both of these have the same DNA sequence in them, why does a spit cell behave so differently than a brain cell? They both have the same identical genome. Okay? And I want to clarify here, when I say genome, I mean the entire collection of DNA sequence within a particular cell. So the whole string of DNA sequences in your cell is collectively referred to as your genome. Okay? And so if the spit cell has the same genetic sequence on the genome as your brain cell, why does it behave differently than a brain cell? Okay? Turns out a lot of this comes down to the packaging. It comes down to how the genome in a spit cell is packaged versus the genome in a brain cell is packaged. In a spit cell, certain parts of the genome are packaged and kept off limits, so they cannot be read. In a brain cell, a different subset of your genome is packaged and kept off limits, so it cannot be read. As a result, the same genome can now code for different behaviors. Okay? And it allows one genome to be differentially packaged, and so in a spit cell, parts of the genome that are off limits are different than parts of the genome that are off limits in a brain cell. Okay? So this is what my lab studies. We study how genome packaging allows a spit cell to be a spit cell versus a brain cell. Okay? Now, before I tell you a little bit about what we've learned about the basics of genome packaging, I want to take a little bit of a tangent and talk about scale. I think there are a few mathematicians, physicists, chemists, and biologists in this audience. Okay? So let's talk a little bit about scale. Okay? If you take the genome from an individual cell, if you take the DNA from a single cell, okay, and if you think of the DNA as a long string, okay, the double helical string, and you stretch it end to end, it's about 6 feet long, as tall as Amitabh Bachchan. Okay? Yet, this long, thin piece of DNA that's as tall as Amitabh Bachchan has to fit into a cell which is 1/100th the width of your hair. Okay? So that's kind of mind-boggling. Okay? And it's the kind of question that drives curiosity-driven research. How is it that this really long, thin but long piece of string can fit into something that is so small? Okay? So, of course, nature has had many years to optimize this. And many people have been studying how this process happens. And we know quite a bit about how this long, thin piece of DNA can be packaged into the small volume of a cell. And so what we know is described in this slide. Okay? So what we know from the work of several folks over the last several decades is that you can start, if you start imagining DNA like so, which is the classic double helical structure, then in human cells and all higher organism cells, the DNA is actually wrapped around spools of protein. Okay? So just as you would wrap a thin piece of thread around a spool to compact and package it, the same kind of solution has been adopted by nature, where you take a long, thin piece of DNA and wrap it around spools of protein. Okay? Now, these spools of protein are then further organized by packaging proteins that condense these spools together and keep them all together off limits. Okay? And so what you have is a mechanism of packaging where you have the first level of packaging where the DNA is wrapped around spools, and then a second level of packaging where these spools are further compacted by packaging proteins. Okay? And so you can see, even if you're not a biologist, you can see that if you want to access a particular piece of the string, if it's tied up in these kinds of spools, you're not going to be able to access the information in that string. Okay? And you can now imagine how, in a spit cell, different parts of the genome are going to be in this state versus this state. If a particular gene is in this state of packaging, it's going to be more accessible to the machinery that reads the information, and the gene is going to be on. If a gene is in this kind of packaging, it's going to be inaccessible because it's going to be covered up, okay? And it's going to be off. And so you can now imagine how a spit cell has different genes here versus here compared to a brain cell. Okay? And so this kind of packaging is therefore very crucial to determine the identity of a cell. Okay? It tells the cell how to behave, it tells the cell how to divide, and so on and so forth. So the identity of a cell depends on its genome, but even at the same time depends on how that genome is packaged. Okay? And so you can then further imagine why it's now logical to see why misfolding of the genome can cause a cell to have an identity crisis. Okay? So if you misfold the genome, now genes that are off are on and genes that are on are off, and now a cell might get confused. Okay? So an ovarian cell will say, I no longer want to behave like my neighbors, I want to become a cancer cell. A brain cell might say, I no longer want to behave like my neighbors, I want to become a brain cancer cell. Okay? And so this kind of identity crisis within a cell has often at its heart a disruption of the correct packaging of the genome. Okay? And so these are the kinds of questions we are interested in. We are interested in understanding how this kind of genome folding happens so we can better understand how misfolding of the genome can cause a cell to have an identity crisis. Okay? And so what are the questions? What are the questions that my group is interested in? So our questions are very fundamental. We would like to understand how large parts of the genome are kept off limits. How is it that they are packaged and kept off limits from being read? Okay? And again, if we go and look into the textbooks, the spools of DNA are described as rigid packaging units. Okay? And that's not surprising. We are humans. We are using our own imagination. If we imagine the spools like we imagine spools that wrap DNA, we would think of them as rigid units, rigid, either rigid plastic spools or rigid wooden spools. Okay? So in textbooks, these spools of protein that are shown in yellow are described as rigid packaging units around which the DNA is wrapped. Okay? At the same time, these packaging proteins, if you imagine you want to sequester a bunch of spools and not have them be accessible, you would imagine perhaps you could build a fortress around them, a rigid fortress that prevents the entry of molecules that want to try and read the information in the spools, that the spools are covering up. And so based on a lot of previous work, in the textbook, the description of these packaging proteins is described as proteins that build a very stable and rigid fortress around the spool DNA. Okay, that keep it locked away, like you would lock some of your jewelry in a safety deposit box. Okay, lock and key, very rigid, very hard to break into. Okay, and so these are the two textbook descriptions of how our genome is packaged. And our questions have been, again, very curiosity-driven and very simple to begin with, but have led us to unexpected answers. So here are our questions. How do these packaging proteins actually do their work? How do they create this fortress? Because if we understand how they create this fortress, we can understand how defects in creating this fortress can lead to disease. And we want to also ask how these packaging proteins act on the spools. Okay? What do they do to the spools when these spools get inside the fortress formed by the packaging proteins? So these are our questions. What is our approach? Okay. So as Kundan mentioned, we use largely biophysical approaches. Okay. And so our approach is to discover the intrinsic biophysical behaviors of genome regulators, like the packaging proteins. So we can imagine, and this is the operative word, imagine and test how these behaviors are used in different cellular contexts. And so we believe that these large packaging proteins and these large biomolecules, or macromolecules as we call them, have personalities. They're capable of doing many different things. And if we can really understand their biophysics, the biophysics behind their personalities, what are all the different things that these macromolecules are capable of doing, then we can imagine and test how these macromolecules might work in different cellular contexts. So it's a little bit like when you're trying to work in a group of people. If you can assess the character of a person, okay, and it's not always easy, but let's say you can assess the character of a person, then you can predict how that person is going to behave in different situations, okay? Because you know what their capabilities are. Okay? The same thing with these complex macromolecules. If you know all of their capabilities, you can understand under one such a situation how this molecule is going to behave versus another set of situations. Okay? And so what I'm going to do next is introduce you to the molecules, and I'm going to try and keep it at a level that hopefully is understandable to non-biologists, and we'll see, but we'll find out at the end when I get questions. Okay, so this is most of the background. Let me introduce you. Let me do a proper job of introducing you to the players, okay, to the molecules. So these are the two molecules we're going to talk about today. Okay. The first is the spool that I was talking about, the primary unit of DNA packaging in human cells. This spool is called a nucleosome, and what the spool has is a collection of proteins that are colored in different colors here. So these are many different proteins that form a spool. Okay? And you can see that the double helical DNA is wrapped around this spool. Okay? And so this is a picture from an X-ray diffraction structure. And you can see why people think of the spool as a very rigid structure. It's nice, it's beautiful, it's complex, and you can see that it really nicely organizes the DNA in about 2 turns. About 150 letters of DNA are wrapped around this. Okay? And we also know that the proteins that form the spool can have different chemical marks on them, can have different chemical modifications on them. And these modifications are recognized by the packaging proteins. Okay? So let's then come to the packaging protein, the main packaging protein that I'll talk about today, which is this protein here called HP1. Okay? Now this protein has, I'm showing you a domain diagram, has 2 parts of the protein that have structures that are folded, and 3 parts of the protein here in blue that seem to be floppy, that don't have structures. And this protein often forms pairs. So you can see one of the partners here and the other partner here, and it forms what we call dimers. So it forms pairs. And so this particular protein is called HP1. I won't give you the history of behind the name. For the sake of the talk, I'm going to just call this protein HP1. But what is important to keep in mind is that this protein and its sequence and its overall structure is conserved all the way from the lowly fission yeast to humans. Okay? So it's an important protein that plays a role in genome packaging across organisms. So we are going to talk about these 2 proteins. And before I talk about our most recent data, let me give you a little bit of background on what we've learned before I jump into the more recent data. So a few years ago, we learned by studying the HP1 protein from yeast that this protein can form long chains that can link multiple spools. Okay? And again, we got to this result through biophysical studies. And what I'm showing you here is a different version of the spool. Again, this is the protein spool. This little black line is the DNA. This is the chemical modification on the proteins that is recognized by the HP1 proteins. Okay. So these are the packaging proteins that are sitting on top of the spool pieces of DNA. Okay. So what we learned from these prior studies was that the packaging proteins that we call HP1 can form long chains, and these chains can then bring multiple spools together and compact them. Okay? And so it's kind of like if you want to bring a group of people together, you can form a chain around them and corral them into a limited amount of space. So it's the same general principle that these packaging proteins, the HP1 packaging proteins, can actually sit across multiple spools and condense these multiple spools together to form a compact structure that's inaccessible. Okay? And so this is what we learned a few years ago, but we had many open questions at this point. How do these HP1 chains actually lead to the compaction and sequestration of chromatin? And so let me make sure I introduce what I mean by chromatin. Chromatin is the general term used for packaged DNA in humans. Okay? And so any form of packaged DNA we call chromatin. And so our general open question was, how do these chains lead to the compaction and sequestering of packaged DNA? And this question was taken up by a very talented and brave graduate student, Adam Larsen. And he said, I want to know how human proteins form chains. We learned how the yeast proteins form chains. I want to learn how the human proteins form chains because humans are more complicated than yeast. Okay? So maybe we learned something more interesting by studying humans. And while he was getting ready to do his studies, he learned from the literature that the human proteins have an interesting quirk. The human HP1 proteins have an extra chemical modification on one of the pieces of the protein that is unstructured, and this is phosphorylation. These are phosphorylation marks on the end, one of the ends of this protein. Okay? And so he said, let me make this protein in a test tube just as it's found in a cell. So I will put these phosphorylation marks on it, purify it, and study its properties. So again, to remind the audience, what we do in the lab is we use biophysical methods, which means that we make pure proteins in a test tube and we study their behaviors. And so he made a very pure protein preparation with this phosphorylation mark. And every time he would purify it, he would come to me and say, things are not working well. Something went wrong. Okay? And Adam is an excellent biochemist. And so it was kind of surprising that he was not able to get this protein to behave. And the reason he would say things are not working is that whenever he made this protein and purified it and made it really pure, this is how the solution would look. Okay? He would phosphorylate the protein, and the solution would become cloudy. Okay? Now, those of you in the audience who have purified proteins know that if you purify a protein and you see a cloudy material, it's usually bad news. That means your protein has precipitated and crashed out of solution. It's misbehaving. Okay? And so when he showed me this, I said, spin it down, take out all the precipitated bad stuff, and take the supernatant, the clear liquid at the top, and study that. Okay? Because this didn't look good. Adam, being a really good graduate student, didn't listen to me. Okay? So instead of listening to me, he took this material and directly, instead of spinning it down or throwing it away, he looked at it under a microscope. Okay? And this is what he saw. Okay? He saw that what we were imagining to be some really messed up precipitate was actually droplets. Okay? And so it was Adam's curiosity about what this material was, and just the simple act of looking under a microscope that led to this first discovery, that this protein can form droplets, and that these droplets, as I will show you later, have interesting properties that can help us explain genome compartmentalization. Okay? And so this turned out to be the beginning of a journey where we learned that these kinds of droplets, which are hallmarks of phase separation, single liquid separating into two components, could be one of the basic principles of genome organization. So after he saw the droplet formation, he went back to ask, how does this protein form chains? Because it turned out that the ability to form chains was essential to form droplets. And what he learned was that this phosphorylation, these phosphate groups, adds a negative charge of the protein, allows this protein to form longer chains. And that's because of another feature of the protein that I didn't mention earlier, which is that one of the pieces of the protein that is unstructured has a bunch of positive charges on it, a bunch of amino acids that are positively charged. And so what Adam learned was that the phosphorylation mark of one pair of proteins would interact with the positively charged residues of another pair of proteins, and this led it to form many long chains. Okay? And the long chains, when they reached a certain critical mass, would then come out of solution as a three-dimensional network of droplets. Okay? And again, he further learned that these were electrostatically driven, and this electrostatically driven process leads to phase separation. So at this point, we were excited, but we were slowly marching our way up to biological relevance. Remember, this is all in a test tube. So we were curious about what this might actually mean for biology. So one of the first questions that Adam asked was, okay, he sees this with this HP1 protein that's phosphorylated. Okay? But we know that this protein also exists in a non-phosphorylated state in cells. Okay? So are we missing something? Is the non-phosphorylated protein capable of forming droplets, or is it not? Okay? And he made a very simple logical connection. He said the non-phosphorylated protein we knew has the same arrangement. It has these positively charged regions, and people had shown before us that these regions can bind DNA. So remember, the role of these proteins is to compact the genome. So they really like to bind DNA. They really like to bind DNA and compact it in one way or the other. And this is the region of the protein that binds the DNA. Okay? Now, even a physicist would know that a DNA backbone has a phosphate, is a phosphate backbone. Okay? And so what Adam made a simple leap. He said, could these effects of these phosphates be mimicked when DNA binds the non-phosphorylated protein? Okay? So if you don't have a phosphorylated protein, but instead bind DNA to it, could you recapitulate this phase separation behavior? And indeed, that's what he saw. What he saw was that, and he learned a little bit more even by doing this. So what he found was that if the protein is not phosphorylated, it doesn't have this little extra chemical mark, it exists in an auto-inhibited and very compact state. And he used a variety of biophysical methods to show that the shape of the molecule is very compact. But when he adds the DNA, he found that the DNA could help open up this protein and organize it and induce this oligomeric state, which is it's just a scientific word for chains of protein, and that this then led to liquid droplets. Okay? So the same phenomena that we saw with phosphorylation could be recapitulated by binding of DNA to these long chains of molecules. Okay? And he could then
labeled the DNA with a fluorescent dye, and he saw that if you looked at these droplets, you can see that the droplets light up with fluorescence. These are droplets made by the packaging protein, and they can fuse and they contain the DNA, and you can see that these droplets are very dynamic. Okay? So these droplets are actually holding the DNA within them, fusing, merging, and so on and so forth. Okay? So this has a very liquid-like property to this organization. It's not this static fortress that people had been imagining in textbooks. Okay? So at this stage, we realized, or Adam realized, he had another opportunity. We know that the DNA is inside the droplets. We know this protein is supposed to help compact the genome. Okay? So was there a way to directly look and see what happens to the DNA itself? Okay? And so for that, we turned to my colleague, Cy Redding, who has been pioneering the use of this single molecule method, where you can directly look at individual pieces of DNA. Okay? So a single molecule method where you can directly look at single molecules of DNA. Okay? And so Madeline Keenan, a joint graduate student between Cy's lab and my lab, did the experiments that I'm about to show you. So let me give you a little brief background on what this technique is. It's quite intuitive. What you do is you take long pieces of DNA and you extract them from a virus that infects bacteria called a lambdaphage. These pieces of DNA are long, intact pieces of DNA that are about 50,000 base pairs. Okay? So 50,000 letters of DNA in the double helix. You can stretch them out. You can attach them to a glass slide. You can on one end, and then add flow, laminar flow that stretches out the individual pieces of DNA. So you're essentially stretching out these long, thin strings of DNA with flow. Okay? So the DNA is attached on one end. You stretch them out, and here you can see the individual strands of DNA stretched out. So you're directly looking at individual strands of DNA here. And these pieces of DNA are lightly labeled with a fluorescent dye so you can see them. Okay? And so these are the DNA curtains. We call them, or Cy calls them DNA curtains. You can see that the DNA is stretched out on the flow. And then what Madeline did was she added some HP1 protein to these curtains. And you'll see what happens. So the DNA is being rapidly and in real time compacted. Okay? And there is no energy in the system. It's all binding energy. It's all the binding energy of these HP1 molecules. And it's happening on a time scale that is more than compatible with what happens in a cell. Okay? And so this was quite exciting to us because remember, what we are trying to do here is uncover the intrinsic biophysical properties of these molecules. Okay? And so what have I told you so far? I've told you that this packaging protein can form chains. Okay? I have told you that this packaging protein, when the chains are long enough, can form droplets that can enclose the DNA. And now I'm telling you that these packaging proteins can compact the genome, pieces of the genome within it. Okay? All of these properties make a lot of sense when you think about the biological context. That is what these proteins are supposed to do. And if we bring all of these three features of these proteins together, we came up with a model, a very simple, intuitive model, where we argued that these HP1 molecules, packaging proteins, can form long chains on the spools of, on nucleosomes, so the spool DNA. And when these chains reach a certain critical concentration, a certain threshold concentration, they can switch into a droplet state that then drags the chromatin, drags these packaged spools of DNA with them. Okay? And so now what you're doing is you're going from an open state of spools to a compartment that's within a liquid droplet. Okay? I also want to mention that our study came out in, it's now 2 years, back to back with the study of Gary Karpin. They came to the same kind of conclusion using a very different set of approaches. And it was really nice to see two different labs coming to the same general conclusion using very independent methods. So both Gary's story and our story together have led to a model like this. Okay? And so let me just spend a few minutes on this model because we also learned, and it's in this published paper, that we could drive this process in both directions by specific ligands, specific proteins that bound the packaging protein, the HP1 protein. So some of the proteins would promote droplet formation that would sit on top of these packaging, and others would dissolve the ligands. Okay? So now we have the beginnings to imagine how this genome packaging can be regulated in the cell, how these liquid droplet-like compartments can be regulated. They can either be formed or dissolved. Now this, when we proposed this model, it caused a lot of excitement because it's simple and intuitive, right? And because what are you doing? You're taking something that is soluble and you're bringing it into a liquid droplet that then prevents other things from getting in. You're shielding it into its own compartment. Okay? And the fact that it's reversible makes it easier for biology to happen. Okay? So now, just to kind of drive home the point, the reason this is intuitive is it's very related to people talking about oil, waters, and droplet. Okay? So when you put oil in water, the two things are not miscible. They don't mix, and you form oil droplets in water. But now imagine, for those of you who cook, right, you're doing tadka, right? And you're putting masalas. You use oil to put the masalas because the masalas are soluble in oil, okay? So if you put oil masalas into a liquid that has water and oil droplets, the masalas are going to go into the droplets. Okay? They're going to partition into the droplets, and the and the oil droplets are going to contain and sequester the masalas from the aqueous phase. Okay? And it's only when you eat and it touches your tongue that you're going to have a burst of flavor. Okay? So it's the same principle that we are proposing for sequestering the genome. Then, of course, it's much more complicated, but at its heart, it's a very similar principle, that we are creating a compartment where the DNA is more likely to partition and be sequestered than outside. Okay? And so this is why this kind of model made a lot of sense and it was interesting. And it also provided a new perspective to target diseases of defective genome packaging. Because when you're designing or thinking about therapies to target these package states, if you know what the package states look like, it's going to be much easier. If you imagine the package states to be these rigid fortresses, then you're going to think of one way of targeting them. But if you imagine them to be these liquid droplets that can be dissolved and formed, you're going to think of a different way of targeting the state. And so this has now provided a new perspective to target diseases in this context, and we and others are working on exploiting this. But we won't talk about that today. I want to talk about the basic curiosity-driven biology and what we learned next. So again, simple model, intuitive. But nothing in biology is simple. Those of you in the audience who are biologists will know. Nothing in biology is simple. You go with a simple model, you go to test it, and you discover nature has put in way more complexity. Okay? And so we asked, okay, for us, this was just the beginning of a new adventure. We asked, sure, this is a good starting point, this is exciting, but can we dig a little deeper? Okay? And can we ask, how is HP1-mediated droplet formation actually related to compacting chromatin? And again, by chromatin, I mean the spooled pieces of DNA. Okay? How is it that this actually works? We can draw cartoons and make anything possible, right? But how does this actually work at the level of molecules? Okay? And so that's the second story that I will share with you today. We wanted to dig deeper and ask, how does the assembly of these HP1 proteins on these spooled pieces of DNA cause the compaction of chromatin, or cause the compaction of the genome? And for this, we decided that we needed to get away from cartoons and give these packaged spools of DNA the respect they deserve. Okay? And what do I mean by that? The packaged spools, remember, look like this. They don't look like these cartoons with black things drawn around it. This is what the structure tells us they look like. Okay? And so our goal was to ask what happens to the structure when we add in one of the HP1 proteins that we study from fission yeast SY6. So I'm not going to use the word SY6 for the rest of the talk, but I just wanted to introduce this for the people in the audience who are biologists. So everything I'm going to tell you now is with the fission yeast HP1. We're going to call it HP1 for the rest of the talk. Okay? And so this challenge of understanding what happens to this structure when HP1 binds was taken up by a very talented postdoctoral fellow, Serena Sanulli, in collaboration with my colleague, John Gross. Okay, so everything I'm going to tell you has been led and done by Serena, and it's a close collaboration with John Gross. And I also want to mention that when the course that we did at IISER was driven in large part, almost exclusively, all the experiments were driven by Serena. So those of you in the audience who've done the course will remember the failed and the unfailed experiments from the course. Okay. So what did Serena decide to ask? She asked a very simple question. When you put a packaging protein on this spool piece of DNA, on the nucleosome, what happens to the spool itself? Okay? What happens to these spool proteins? Okay? Now, if you think from first principles, what is the function of these packaging proteins thought to be? They cover up the spools and they make them inaccessible. Okay? So the simplest expectation from Serena's experiments was that if we added the HP1 protein on top of this nucleosome, these proteins, the spool would become less accessible to solution. It would be covered up. Okay? That was the simplest expectation. Okay? And so what that means in biophysical terms is that binding of this HP1 protein would reduce the dynamics of the structure, would make it more rigid, and would reduce the accessibility of these proteins, these spool proteins, to the solvent. Okay? And so Serena did an experiment, a classic biophysical experiment, that directly allows you to look at the solvent accessibility of any protein that you want to study. Okay? And she focused on looking at the solvent accessibility of the spool proteins when HP1 binds on top. Okay? It's a very simple premise, but a complicated experiment. And what's the premise of this experiment? She takes the spool, the nucleosome, she adds the HP1 protein, and she puts it, instead of in H2O, she puts it in D2O. Okay? And what we know from work of several folks before us is that the protein backbone has an amide bond, and the proton in that amide bond exchanges very readily with the solvent. And so if you put it in D2O, the proton will exchange for a deuteron. Okay? But the amount that it exchanges with the solvent depends on how accessible it is to the solvent. Okay? And so what you can ask is, in this structure, if you just put it in D2O over various amounts of time, how much of the deuterium gets incorporated inside the protein, inside the spool protein? Okay? And based on the structure of the spool, different parts of the spool are going to pick up different deuterium. The parts of the spool that are more buried inside are going to pick up less deuterium because it's going to have a harder time getting in. And the parts that are on the surface that are near the solvent are going to pick up more deuterium. Okay? Very simple premise. So what Serena asked is, what happens to the deuterium uptake across the spool proteins in the presence of HP1? Okay? When you add HP1, what happens to deuterium uptake? Again, the simplest expectation was that when you add the HP1 protein, it would become less solvent accessible, so that would mean less deuterium would be taken up overall. Simple premise. I think people are shaking their heads, you get it, right? And so the way the experiment is done, you mix these two proteins with D2O for different amounts of time. You quench the exchange by lowering the pH, because when you lower the pH, you lower the exchange. You digest with a protease that chops up the pieces of the spool, and then you do mass spec, and which tells you chop up the protein and you see how different pieces of the protein picked up deuterium. Okay? And so when Serena did the experiment, she got the exact opposite result of what she predicted. Okay? Instead of less deuterium being picked up when HP1 is bound, there was a widespread increase in deuterium uptake throughout the nucleosome. Okay? And so what I'm showing you here is a slightly different representation of the spool proteins. So there are 4 proteins, and again, the names are not important, but they're here for consistency. There are 4 proteins, H2A, H2B, H3, and H4, that come together to make this spool. Okay? And so what Serena found was that each of these proteins in this structure picked up a lot of deuterium when HP1 was bound compared to when no HP1 was bound. Okay? And the bluer the particular region of the protein is, the more deuterium it's picked up. Okay? And you can color code this on top of the nucleosome structure. Okay? I've only color coded half the nucleosome. It's a pseudo 2-fold symmetric structure because there are 2 copies of each of these. And what you can see is that there is dark blue almost throughout the whole spool protein. Okay? And Serena then did something else. She asked which regions of this spool picked up the highest amount of deuterium, showed the biggest differential in picking up deuterium. And she color-coded them so that we could more easily represent this. And shown here is a different version of the data, where in orange are the regions of the nucleosome or the packaging, of the spool proteins that are normally occluded by DNA. So the DNA is wrapping around it, so they're occluded by DNA. And these pick up a lot of deuterium. And the second part of the protein that picks up a lot of deuterium are regions of the protein that are occluded by interactions between the spool proteins. Okay? And so to show you that these regions are actually buried in the canonical structure, in the structure seen in textbooks, here's a space-filling representation of the same nucleosome right here. Okay? And you can see the red and the orange you can barely see. Right? And you can imagine a deuterium molecule, D2O trying to get in, is going to have a hard time getting in through the nooks and crannies. Okay? And so somehow, what the data was telling is that somehow the HB1 proteins, they're sitting on the top of the nucleosome and are unfolding the core of the nucleosome and making it accessible to pick up deuterium from solvent. Okay? Now, this was pretty bizarre, right? Because remember, again, the role of HB1 proteins is to reduce access to chromatin in cells. But at the level of the smallest packaging units, these proteins are sitting on the top and making the spool itself much more accessible. It was opening it up so that the spool could pick up deuterium. And so because it was such an unusual result, of course, we tested it with multiple methods, including a specific type of spectroscopic method called methyltrosy NMR. I'm happy to discuss this with people later on. But even testing by complementary method, we saw a very similar result. We saw that pieces of the spool inside that were buried were showing increased dynamics. And these residues were hydrophobic residues, or they're residues that like to stay away from water, which is why they're buried inside the core of the spool. But even these buried hydrophobic residues were showing a higher number of dynamics when the HB1 protein bound. Okay? And it also told us that, the NMR experiment also told us that the nucleosomes don't fall apart. The structure remains intact. Okay? So by this point, we were convinced that what we were seeing was real. There was not an artifact of one type of experiment. We had done these two experiments, the hydrogen deuterium exchange, NMR, and one more experiment that I'm not talking about. And we found all three experiments gave the same result, that binding of the HB1 protein on the structure opened it up rather than shutting it down, okay, and made it more dynamic. And so then we asked, how is this protein doing it? And we asked this question by asking, how does this protein actually engage the DNA spool, the spool DNA? And we did cross-linking mass spectrometry, a variety of different methods, and I'm going to not talk about the details of the methods, but using a variety of methods, Serena, in collaboration with Mike Trinka and Al Burlingame, was able to figure out how this protein actually touches the nucleosome. And what she learned was that this protein binds a piece of the folded spool here and unfolds it. It also binds this chemical modification on the spool, and it can also bind the DNA that's wrapped around the spool. Okay? So the DNA itself that is organized in the spool. And that we knew from before, because remember, this is the same hinge that binds DNA that drives phase separation. Okay? And so putting together data from a variety of different methods, what Serena was able to propose is a model where two pairs of these molecules bind a spool and hold it in multiple places. They engage one of the pieces of the spool that has a chemical modification in one particular location that orients this HB1 molecule on the spool. The hinge piece that has the positively charged residues binds the nucleosomal DNA, and this little piece here that we call the CST binds a piece of the spool itself and unfolds it. Okay? So now what we have is a structure where the HB1 molecule is hugging the nucleosome from outside, okay, and doing three things simultaneously. It's recognizing this chemical mark, it's binding the DNA and loosening the grip of the DNA on the histone, on the spools. I keep saying histones. Histones are the names of the spool proteins. And this little part of the HB1 protein is gently unfolding part of the spool. Okay? So the consequence of all of these interactions is that you are loosening the interaction of the DNA with the spool and allowing the proteins in the spool to breathe more. Okay? So it's very analogous to those of you who wear collars. If you loosen your collar, you can breathe better. What we think these HB1 proteins are doing is loosening the DNA collar around the spool. So now the spool, instead of having to be rigid, is dynamic and flexible and can dance and can open up and can expose its true self that is buried inside. Okay? And so at this point, we'd figured out that this happens, and we'd figured out how it happens. And that led to the question of, why it happens, right? Why would you have such a paradoxical function for a protein that is supposed to shut down chromatin? Okay. And so we wanted to ask, is this phenomena that we're seeing at the level of the smallest unit of DNA packaging, does it have anything to do with higher order chromatin organization, compaction of the genome at a higher level? And so for this, what Serena did was she adapted a very old assay, where instead of looking at single spools or single nucleosomes, she used strings of nucleosomes. Okay? So now we are moving from the smallest unit to a string of nucleosomes that is more like what you might see in parts of the genome, where each of these spools now has a piece of the DNA wrapped around it. You have a piece that's not, it continues, and so on. So it's one continuous piece of DNA wrapped around 12 spools. Okay? And she asked what happens when she puts this HP1 protein with the spools using an assay that looks at condensation of these arrays of spools together. Okay? And remember, we are trying to look at the ability of this protein to bring parts of chromatin together and condense it. Okay? And so what she did, again, is a very simple assay. You mix this protein with this string of spools, and you spin it down. If HP1 allows these strings, long strings of nucleosomes to come together and compact, they would precipitate, and you would see a depletion of signal in the supernatant, in the clear liquid. Okay? And this assay has been used in many other contexts with human proteins by other groups. Okay? And so what you see here is what Serena got. These are long strings of nucleosomes, again, that have the specific chemical mark that is recognized by this packaging, by the HP1 protein. Here's the signal of the unprecipitated chromatin here. And as you increase HP1 concentration, less and less of it is in solution, more and more of it gets precipitated. Okay, so this was not surprising. This is expected because this protein is supposed to bring chromatin together and precipitate it. Okay. So now we had an assay in hand. What Serena asked was, what is the role of these dynamics? What is the role of these increased dynamics caused by the protein? Oops. My computer is showing some unexpected dynamics. Okay, let's go here. So the way she tested the role of the dynamics was by using a trick. Oops. Yeah. That we can take advantage of by making a specific cross-link within the spool of the protein, or spool of the nucleosome. And this is because none of the spool proteins, none of the histones that make the spool have any cysteines. Now, cysteines are special because you can make disulfide bonds with cysteines. Okay? And so what Serena did was she introduced 2 cysteine residues at location within the spool and formed a disulfide bond between them that would constrain the interface. Okay? So that would prevent the dynamics that were caused by the HP1 protein. That would lock this interface together and restrict the dynamics that are caused by the HP1 protein. And she then asked, if she restricts these dynamics, what happens to the ability of HP1 proteins to condense chromatin? And what she saw is shown here by the green plot, and hopefully you can see it in the back. When she uses long strings of nucleosomes that now are constrained, even just at one interface that we saw become more mobile by deuterium exchange, she sees an inhibition of the ability of this protein to precipitate these arrays. Okay? So somehow, the increased dynamics in the core of the individual nucleosomes was important for allowing chains of nucleosomes to come together. Okay? So again, we can now say that this is important, but we still need to understand why it's important. Okay? So clearly, what HP1 is doing at the level of a single nucleosome has an effect at the level of organizing large stretches of chromatin. Okay? And she then did something that everybody in my lab now routinely does after Adam's experience. So this is a really old experiment, or old approach, where you precipitate. Okay? And in the old days, people would take the precipitate and look at it on EM grids. Okay? But what Serena did was do exactly what Adam did. She looked at it in a microscope. She did not fix it. And she saw droplets. Okay? So we've now come a full circle. Okay? So now what we're seeing is that in the presence of chromatin, what people have been talking about as a chromatin compaction assay is actually a droplet formation assay. Okay? And so as you increase the HP1 concentration, when we see chromatin compaction, when you see precipitation of this chromatin, what we are actually seeing is formation of droplets. And so if you prevent dynamics within the core of the nucleosome, then you're not just preventing precipitation, you're also inhibiting droplet formation. Okay? And so for us, this was exciting because this was our first direct evidence that chromatin organization and compaction and phase separation into droplets are directly coupled. Okay? And again, now we've directly tested part of the original model that chromatin itself, when it gets compacted, forms these phase separated droplets. Okay? It's not just HP1 passively dragging chromatin inside the droplets. Okay? So at this point, I've shown you a lot of data. I'm going to spend the next few slides just speculating about what this all means. Okay. So the big question that we had was, why would exposure of the histone core, and when I say the histone core, that's the core of the spool, promote higher order chromatin organization? Again, it's non-intuitive, right? Because if you're compacting chromatin, you want things to be more occluded. So why would exposure of the histone core promote higher order chromatin organization? And let's first look back at the textbooks. Okay? So there's a lot of good work done on understanding how chromatin or nucleosomes are organized together to form a compacted chromatin. And what is learned so far is that if you look at the structure of the nucleosome, there are these histone proteins that form the spools. The histones have a lot of unstructured regions that stick out from the spools. Okay? And these turn out to be positively charged. So what people have found is that these positively charged tails of one nucleosome can interact with the negatively charged DNA of a nearby nucleosome and help compact or bring these structures together and form more condensed states. Okay? And so this is known, and there's a lot of data to support this. And so just from first principles, if you're compacting a long string, you want something that mediates interactions between the individual residues of that string. So you want something that mediates interactions between this nucleosome and so on and so forth. So in this kind of a model, if you now imagine, if you now have increased exposure of the buried histone residues, you're bringing on deck additional interfaces to make internucleosomal interactions. Okay? So you don't no longer have to rely on just the histone tails interacting with the DNA, but when you expose the hydrophobic core of a nucleosome, you're now bringing on deck the possibility of using hydrophobic interactions to form internucleosomal interactions. Okay? And so this is what we propose. We propose that what HP1 is doing is it's increasing the opportunities for internucleosomal interactions, which is increasing the opportunities for how these nucleosomes interact with each other. Now, these are very transient interactions, okay, because what HP1 does to a nucleosome is it transiently opens it up and unfolds it. Okay? So why would this be important? Well, let's come back to a simple version of the model. Okay? So what we would like to propose is that HP1 proteins act as nucleosome disorganizing agents to transiently expose buried histone residues. And so where this leads us is the concept that here's how a nucleosome or a spool looks when you look at a crystal structure. This is the thermodynamically most stable state of a nucleosome. Okay? What we would like to propose that this is just one of many conformations that this spool can adopt. That this spool really has the capability of adopting multiple conformational states, okay? Shown here by an ensemble of additional conformations. And it is these additional ensemble of conformations that is where a lot of the biology comes about, where these spools no longer just act as packaging proteins, but directly participate in making interactions with other proteins, like other nucleosomes or with histone, with HP1 proteins. And so what we propose HP1 proteins are doing is they're unlocking the potential of a nucleosome. Okay? They're unlocking the potential of a nucleosome by making these additional conformations accessible under physiological conditions, under physiological temperatures and buffer conditions. And so what we would like to propose is that the nucleosomes are not just boring packaging materials, but coming back to the way we do experiments in my lab, these nucleosomes have a personality, and their real personality is hidden. It's buried inside this structure. It's only when a molecule like HP1 comes along and loosens the structure that the real potential of these packaging units becomes possible. Okay? And again, there are many other labs that are finding evidence that a nucleosome doesn't just adopt this state, but it can adopt different kinds of conformational states. And so what does it mean in terms of HP1-mediated chromatin packaging and higher order states for occluding chromatin? What we think is that transient exposure of the buried histone residues increases options for weak multivalent interactions that promote phase separation and occlusion of chromatin. Okay? So in the end, the paradox is resolved because these exposed residues participate in other interactions, and the end product is a phase-separated state that occludes the chromatin. Okay? So we started with the paradox that you're opening up a nucleosome, but we're ending with a resolution because the opened up nucleosome is important for participating in other interactions that will eventually shut down, will eventually package this system into a occluded phase, a phase-separated state. Okay? And studies in many different phase separation systems have shown that what drives phase separation is multivalent interactions. One molecule, that means one molecule being able to make multiple interactions, and that these multivalent interactions be weak and dynamic. Okay? And so what we are proposing is that what HP1 does to nucleosomes is open up the guts of the nucleosomes in a transient way that allows them to participate in weak and dynamic interactions. But because you have a string of nucleosomes, you also get multivalency. Okay? And finally, we'd like to argue that chromatin is not a passive occupant of HP1 droplets. It's not just being dragged into these droplets, but it's actively participating in the process of chromatin compaction. And while I've shown you that this kind of phenomena of altering the packaging unit holds in the context of packaging proteins forming higher order compacted states, what we'd like to propose is that beyond higher order packaging, once we accept the fact that a nucleosome can adopt other conformational states that can be exposed by the action of other proteins, then there's a whole world out there waiting to be discovered. Right? If a nucleosome can do this, then nature must be using these alternative conformational states. And you can begin to imagine that similar mechanisms may generally regulate higher order chromatin compaction, even compaction not driven by HP1 proteins. And if you change the sequence of the spool proteins, the histones, sequence changes might alter the equilibrium for forming this ensemble, and chemical modifications on the histone proteins might also directly affect the deformability and the ensemble adopted by the nucleosome. And to this note, there's a recent paper by the Muir and Alice labs that have found mutations in the core, mutations in these proteins that are associated with cancers, and many of these oncohistone mutations are in the core of the histone protein. Okay? And so it's kind of tempting to speculate that perhaps these oncomutants are altering this equilibrium and in that way causing diseases of misfolding of the genome because you're going to affect higher order chromatin. So I'm going to just end with 2 slides. So I want to come back to where we started, which is our questions, which is how are large parts of the genome kept off limits? When we began, this was the textbook description for the spools, that the spools are rigid packaging units, that the packaging proteins form a fortress. And of course, we know that misfolding can cause the cell to have an identity crisis. And what we've learned over the years is that it is not so. And I'm sure there'll be more to learn, and perhaps our models will also be disproved. So this is just a work in progress. What we have learned that the packaging proteins do not form a rigid fortress, but instead they form dynamic membraneless liquid compartments. And we found that the spools are not these rigid packaging units, but they are shape-shifting participants in the process, that they can adopt multiple conformations, multiple shapes. And what is exciting about these findings is that these kinds of descriptions of the behavior of the packaged genome can help us explain the responsiveness of the genome to developmental signals. If something is a dynamic membraneless liquid, a signal can cause it to dissolve rapidly or it can cause it to form rapidly. So we can begin to now ask how do these things work in the first place in response to signal, and how do these things go wrong? And again, as I mentioned in the beginning, it's given us new avenues to target defects in chromatin organization. So I want to end by thanking all the people who were very important and were the crux for this work. I want to thank first the heroes and heroines of the scientific work, Serena Sanuli, Adam Larsen, Madeline Keenan, and our wonderful collaborators. Adam Larsen got us into this whole area by following his curiosity. Serena got us into learning about the dynamic nature of nucleosomes by not being fazed by getting a result that did not make sense. Okay? And so these are really the heroines and heroes of the work. Also, in addition to our collaborators, I also want to thank Alok Srivastava, our resident philosopher, historian, and sociologist of science, for helping me see the real significance of our work in the context of both history and the broader context of biology. and these people for funding. Thank you.